Looks like we are leveling off a little bit for the flurry of attendees. So welcome again. I am David Leibovitz, a professor of medicine here at Northwestern, and it's my pleasure to help tee off our latest Northwestern Healthcare Artificial Intelligence Forum, which is sponsored through our Center for Collaborative Artificial Intelligence in Healthcare, which is part of our Institute for Artificial Intelligence in Medicine. And it's my distinct pleasure today to be able to introduce our guest speaker for today, Rekha Sathian, who is a PhD student in the Department of Biomedical Informatics at Stony Brook University. And we appreciate Stony Brook University's participation in these sessions as well. And today's topic will be genomic language modeling for predicting enhancers and their allele-specific activity in the human genome. So exciting, late-breaking information about genomic language models. Rekha Sathian's research in general focuses on decoding the regulatory language of the genome using large language models such as DNABERT. She developed DNABERT-Enhancer, a genomic language model trained on the human genome to identify and characterize enhancer regions based on their underlying DNA sequence patterns. Her work aims to elucidate how sequence context governs enhancer activity and how allele-specific variations in non-coding regions can lead to gain or loss of function regulatory effects, advancing your understanding of the genetic basis of complex diseases. Rekha Sathian works under the supervision of our colleague, Dr. Ramana Davuluri. Alongside her research, Rekha is actively involved in next-generation sequencing data analysis and contributes to the design and implementation of bioinformatics pipelines for both clinical and research applications. Before joining Stony Brook, she worked as a senior bioinformatics analyst at MedGenome Labs in India, where she led the development of next-generation sequencing analysis workflows and data-driven solutions. She holds a master's degree in bioinformatics and a bachelor's degree in zoology from Mahatma Gandhi University in India. So thank you so much, Rekha, and I'll let you take it away.
Oh, thank you for the kind introduction, Dr. Leibovitz. Hello, everyone. I am Rekha, a 15-year PhD student doing research under Dr. Davuluri from Department of Biomedical Informatics at Stony Brook University. Today, I'll be talking about how genomic language models can help us understand how non-coding DNA regulates gene and how small genetic changes can alter this regulation. So recent advances in AI are transforming how we study biology. Models that were initially designed to understand human languages can now learn the patterns in the DNA in much similar ways, treating the genome as a language with its own rules. So this intersection of biology and AI is now opening door to uncover how genes are controlled and how tiny differences can influence diseases. The insights which were possible even a few years ago. So when we look at the human genome, only about 2% actually codes for protein. The remaining 98% is non-coding, but that doesn't mean it is not important. In fact, much of this non-coding DNA contains key regulatory elements that control when and where genes are expressed. Enhancers are like one major class of these dysregulatory elements, and they are relatively short, usually around 100 to 1,500 base pairs, but they make up a substantial fraction of the non-coding genome. Even though they don't code for protein, but they play a powerful role in controlling transcription. Enhancers work by boosting the activity of their target genes, even through long-range interactions. Now, how the regulation happens. For an enhancer to become active, pioneer transcription factors first bind to the DNA, then open up the local chromatin environment. This makes the region accessible for additional proteins such as transcription factors, co-activators, chromatin remodelers, and even RNA polymerase II. So together, these proteins prime the enhancer for the activation. So once they are active, the enhancer loop over to its target promoter and helps assemble this protein complex, which is also known as pre-initiation complex, which ultimately increases the transcription of the gene. Beyond their functional roles in gene regulation, enhancers also carry significant clinical relevance. When they malfunction, they directly contribute to human diseases, which are collectively known as enhanceropathies. This disruption can come from many directions. The most common one is the small non-coding mutation, or what you call the short variants, within this enhancer region. Some of the examples I have listed out, which are recently published. The Wang et al. Study in 2023 in Genome Biology, they systematically screened enhancer mutations in melanoma and showed that even single base pair changes in enhancer can significantly alter the gene expression and contribute to tumorigenic fitness. Another study published in the same year, they used comparative genomics, epigenomic data, and a deep learning model to identify approximately 4,000 new enhancers in humans that likely rose from single nucleotide mutations. The authors linked these gain enhancers to transcription factor changes and also genes involved in the neural development and diseases. Another 2023 study showed that SNVs can increase transcription factor binding affinity in an enhancer, which is responsible for GATA4, driving a gain of function expression and cause cell developmental defects. In a recent study in 2024, the authors identified 135 enhancers in breast cancer that carry somatic mutations that correlate with changes in chromatin accessibility, and some of these mutated enhancers are associated with prognosis in specific breast cancer subtypes. So even subtle alterations in enhancer sequences or even the chromatin changes can misregulate key regions. The second mechanism is a structural rearrangement that can literally move enhancers to the wrong genomic context. So it is basically called as enhancer hijacking. This is one of the classic examples in a published study in 2023 in Blood Journal. And the CRISPR experiments in the study validated that a BCL11B enhancer, which is moved to TLX3 oncogene, and it basically hijacks the TLX3 expression and driving high genomic oncogenic expression. And the third mechanism involves the spatial genomic architecture, which is also known as the TAD disruption. A 2024 study published in Nature Communication showed that how deletion of a TAD boundary caused the fusion of two domains, adjacent domains, and allowed the ectopic gene enhancer interactions. So these examples highlight that disruption to enhancer function, whether it is through a single level mutation or even epigenetic changes or genome architecture, they can misregulate transcription in very disease-driving ways. This is why understanding enhancer logic is central to interpreting disease-associated variants, especially that falls in the non-coding regions. Genome-wide association studies are a comprehensive repository of such SNPs which are associated with traits. What this catalog highlights is a sheer scale of genetic associations accumulated across thousands of publications. But the real question or the key point is where these variants actually lie in the genome. When we categorize the GWAS SNPs by genomic annotation, we see that an overwhelming majority of disease-associated variants are non-coding. About 59% of falls in the intron, followed by another 25 lies in the intergenic regions. And only a very small fraction are coding. And you can also see a significant portion mapped to regulatory regions, approximately here. Approximately 5% falls in the annotated regulatory regions. So it is yet to unravel like how many more regulatory associated SNPs, especially the enhancer associated SNPs, are hidden in this large pool of non-coding variants. So this is mainly due to the challenges in basic enhancer biology. It is unclear which gene they target and what tissue they are active. Now that we have seen how critical enhancers are in driving diseases, let's discuss the therapeutic opportunities it brings. One powerful strategy is enhancer-focused gene editing. A landmark example is the recently FDA-approved CRISPR-Cas9 therapy for sickle cell disease and beta thalassemia. So here in the study, rather than correcting the causal mutation, the therapy disrupts a repressor element within BCL11A enhancer. So by lifting this repression, the treatment reactivates fetal hemoglobin, which compensate for the defective ones. So this represents one of the first and most compelling demonstration that editing an enhancer itself, rather than the causal gene, can effectively cure a genetic disease in humans. The second therapeutic avenue is targeted small molecule modulation of enhancer activity. So instead of editing the genome here, these therapies work by disrupting the transcriptional machinery that the super enhancers rely on, which is the causes of cancer. I have listed two studies here. One is GNE 987, which is a BRD4 degrading PROTAC. They act by collapsing the oncogenic superenhancers and selectively eliminates cancer cells that are dependent on this regulatory hubs. Similarly, there is a CDK7 inhibitor which has been found, T8Z1. It suppresses the transcription initiation and silences superenhancer-driven oncogenes such as MYC. Together, these examples shows that enhancers are no longer passive regulatory annotations that we study. They are emerging as actionable therapeutic targets, opening an entirely new landscape for precision medicine. Now, when we think about why enhancer discovery is still such a difficult problem, it really comes down to two major limitations: the complexity of enhancer biology and the constraints of the experimental methods we use to study them. When we focus on the general challenging features, we see that enhancers have several intrinsic features that prevent us from defining them cleanly. They vary widely in length and often have ambiguous boundaries. They can function in either orientation and are highly context dependent. Their distance to target genes varies. It spans from maybe a few kilobases to megabases, making it hard to predict enhancer gene relationship. And the most importantly, enhancers lack a universal sequence code or a consistent set of genetic or epigenetic markers that define them across all biological contexts. So these features make enhancer fundamentally hard to pinpoint and characterize. Coming to the experimental challenges, the experimental methods we rely on introduce their own challenges. Many enhancer identification methods are indirect and depending mainly on markers like histone modification or chromatin accessibility. And the functional assays, whether they are reporter-based or CRISPR-based, they remain low throughput, expensive, and labor-intensive. Their outcomes depend heavily on cell type or specific experimental conditions, and scaling these assays across the entire genome is still difficult. And even after identifying an enhancer, linking it to the correct target gene remain a persistent challenge. So these biological complexities and the methodological limitations have kept enhancer discovery and its interpretation from reaching the level of accuracy and completeness we need today. And this limitation has given rise to the development of a flurry of computational methods or AI models for the identification of enhancers. The models developed over the period of time mainly falls into three categories. First one is feature-based machine learning models, which are the earliest AI approaches. They can be divided into mainly two types. One is chromatin-based models, which uses features like histone marks or other epigenetic signals as features, and they provide indirect clues about the enhancer locations. The second category is the sequence feature models. So they rely on handcrafted sequence features or even nucleotide compositions. So this definitely improved prediction accuracy compared to the previous models, but they remain limited by feature engineering and short sequence contexts. Second is we have deep learning models, which mostly uses the conventional neural networks. And it gained popularity as CNN automatically learn hierarchical sequence pattern without manual feature engineering. And there were some hybrid methods also came, which combine CNN with other modules to capture long-range dependencies and rich context. And finally, we have NLP-inspired models that leverage transformers or pre-trained language models. And these models benefit from contextual embeddings learned from large-scale DNA sequences, but their classification layers remain relatively lightweight, and training still depends on labeled dataset. So unlike the methods we mentioned about the NLP-inspired models that require an additional classification layer, standalone genomic language models encode regulatory information inherently. In this model, the DNA sequence is treated like a language where nucleotides or the k-mers acts as a token, and the surrounding sequence provide the context, much like how words are in a sentence. So these tokens are fed into transformer models, which uses the self-attention layers to learn the relationships across the sequence. The model generates contextual embeddings that capture dependencies between motifs, subtle variations, and even long-range interactions in the genome. Importantly, these embeddings can be used directly for downstream tasks without requiring an additional handcrafted feature layer or additional models. So this approach allows the model to generalize across tissue, species, and cellular context. However, the challenge remains. One is the interpretability is difficult, and the second, we still need robust experimental validations to ensure the model captures true biological signals. Overall, this represents a shift from traditional feature engineering to learning the regulatory syntax directly from the genome. So, building on the idea of treating DNA as language, DNABERT is one of the first widely recognized transformer-based language models, specifically trained for DNA sequences. The model is pre-trained on large genomic corpora using cell-supervised objective. So it learns nucleotide patterns and context without any label data. So coming to its architecture, the DNABERT first splits DNA sequences into overlapping k-mers, which acts like a token in language model. And each k-mer is mapped into a dense embedding vectors, capturing its identity. And these embeddings are then fed into a stack of transformer layers where self-attention allows the model to learn the dependencies between the k-mers, even if they are far apart in the sequence. The output is contextual embedding that capture both local motifs and long-range dependencies across the genome. And finally, these embeddings of the DNABERT can be used for fine-tuning for downstream tasks. Now, over the last few years, several extensions of the original DNABERT has been developed, and the most important one is the DNABERT-2, which is pre-trained on genomes from 135 species. And the main difference is that instead of k-MERs, here the DNA is tokenized by byte pair encodings. Another one was DNABERT-S, which is published later in 2025 by the same author. It is built on DNABERT-2 but integrates species-aware embeddings and undergone enhanced pre-training on diverse genomic data. This model was mainly used for sequence classification and motif detection. Later, recently, we have another model developed, which is pre-trained on huge viral genome, which is termed as HVILM, and they mainly focus on prediction tasks like pathogenicity classification, transmissibility assessment, etc. And another model, pre-trained model, is DNABERT-MP, and which is pre-trained on human microbiome-derived genomic sequences. Now, apart from the pre-trained model, we have fine-tuned models from the DNABERT which had shown top-tier performances in the classification or prediction task like promoter models, ChIP-seq transcription factor binding site models, splice site models. And recently we published it's under review. It's a model called DeepView, which has a group of models, approximately 700 ChIP-seq data has been used for this. And they predict functionally disruptive variants. And then we have tissue-specific promoter models. So although many machine learning models developed so far for the prediction of enhancers, they perform reasonably well, but they often fail to fully capture the sequence context of regulatory regions. This led to suboptimal predictions. And DNABERT, however, has demonstrated top-tier performance in interpreting elements, especially within the non-coding regions, including, as you can see from this slide, promoters, splice sites, and even transcription factor binding regions. So we hypothesize that leveraging the pre-trained DNABERT model to build a specialized enhancer prediction framework can help uncover the nuanced regulatory roles of enhancers in health and disease. And this can contribute to better clinical outcome. So we developed DNABERT-Enhancer, a fine-tuned model built on the pre-trained DNABERT Genome Foundation model, and which effectively captures the contextual and semantic informations of DNA in human genome. In this study, we show that DNABERT-Enhancer can learn the contextual patterns of enhancer regions directly from the sequence alone by leveraging the attention mechanism to highlight biologically meaningful sequence features. The foundation of our dataset comes from the ENCODE consortium. The ENCODE encyclopedia serves as one of the most comprehensive resources for mapping functional elements in human genome. ENCODE integrates large-scale experimental assays through uniform pipelines to generate consistent, high-quality annotations. One of the major output of this effort is the registry of candidate cis-regulatory elements, and these are accessible through Screen Portal. We curated enhancer elements from the ENCODE version 3. They are anchored mainly on DNase-1 hypersensitivity sites, which are usually ranging from the length of 150 to 350 base pair. And they consist of high resolution mapping of 961,000 enhancers. So from the length distribution, we observed two clear peaks around 201 base pairs and 350 base pairs. This natural modes of enhancer length guided our decision to construct two datasets at these fixed length for fine-tuning. Overall, ENCODE's high-confidence enhancer annotation forms the primary positive set for our training data and thereby ensure that the model learns from well-supported regulatory regions. So, In addition to the ENCODE CCRE enhancers, we incorporated enhancer regions from several high-confidence experimental databases and from prediction-based databases. So while we did not directly use them as positive labels, they were extremely important for defining the boundaries of the regulatory regions across the genome. And this ensured that the negative instances represent true non-enhancer regions rather than unlabeled or weak regulatory elements. For the negative set, we sampled the regions of human reference genome that did not overlap with any known or predicted enhancers from these combined sources. This step was crucial for reducing label noises and improving the reliability of the training data. We maintained a 1:1 ratio between both class sequences in both the data sets. Together, this strategy allowed us to build a clean, balanced, and biologically grounded dataset that the model could learn from effectively. Now, using this two dataset, we fine-tuned the pre-trained DNABERT model, resulting in two fine-tuned models, which is named as DNABERT-Enhancer 201 and DNABERT-Enhancer 350. In terms of the predictive power, the 350 base pair model performed the best, achieving an accuracy of 88%, as you can see from the figure A, with an MCC of 76%, while the 201 base pair model reached an accuracy of 82%. This performance difference is likely reflects the additional contextual information available in the longer enhancer sequences compared to the 201 base pair. We also wanted to understand how robust the model is to different choices of negative sets. So we trained two additional variants of our best model, which is the 350 base pair model. So first we created a refined model where the negative sequences were drawn from random genomic regions, but by only excluding the high confidence enhancer set. This model, which we named as DNABERT-Enhancer 350 refined, performed almost identical to the original one with an accuracy of 87.8%. And this suggests that the original model is not overly dependent on weak regulatory features in the background. It is indeed learning enhancer specific sequence patterns. And the second, we trained a harder version of the problem by limiting the negative set to DNA's one hypersensitivity site, which do not overlap with any of the non-enhancers. This model showed substantially lower performance with an accuracy of 61.5%. This reflects that the biological challenge of distinguishing enhancers from other open chromatin region. However, it is also important to note that DHS annotations evolve over the year. For example, more than 11,000 DHS labeled as non-enhancer in screen data version 3 as of 2022 are now classified as enhancer in screen version 2025. And among these updated regions, we evaluated for those with the 350 base pair using our model, and our model predicted 86% of them as the enhancers. So this supports the idea that some DHSs previously labeled as non-enhancer may indeed be true enhancers that were just not annotated at that time. We also benchmarked DNABERT-Enhancer against both classical machine learning models, the traditional ones, and the latest enhancer prediction methods. Our model clearly outperformed all traditional classifiers, which struggle particularly with identifying the true enhancer sequences. By using the DNABERT embedding, we ensured that a fair comparison is done across all approaches. When compared with the recent state-of-the-art method, DNABERT-Enhancer performed well by maintaining high accuracy, F1 score, and specifically MCC scores, regardless of the length of the data we used. Now, we further evaluated the robustness of our model by applying DNABERT-Enhancer 350 across the entire human genome, which led to the identification of approximately 1.82 million enhancers of varying length, and it collectively spans over 21% of the genome. Since our model is trained only on 5% of the um, uh, screen data, we compared our prediction to its entire collection, and we observed that our prediction recovered nearly 79% of all enhancer annotation. And the remaining predictions were further evaluated using multiple public enhancer databases, and a large portion of them intersected with at least one external resource, thereby validating almost 92% of the predicted enhancers. Because the attention patterns laid by DNABERT-Enhancer do not rely on any fixed positional features, our predictions are partially overlapped with the database annotations. We also assessed how well our genome-wide predictions align with the independent enhancer resources separately, and the results were interesting, and notably the one with the Vista enhancers, which has nearly all experimentally validated enhancers in that catalog, and our DNABERT-Enhancer 350 successfully recurred nearly all of them and showed a substantial agreement with the enhancer sets from Enhancer Atlas and typical enhancers from the SCDB, including many large enhancer segments and portions of super enhancers. To ensure these overlaps weren't due to any broad genomic span, we conducted permutation tests, and all datasets showed strong enrichment with highly significant p-values. Altogether, this confirmed that our predictions are strongly supported by external evidence and also reflects the genuine enhancer biology across the genome. Our approach of merging the consecutive enhancer windows while prediction identified several unusually long enhancer regions that exceeds 30 kb, and these broad segments map to multiple enhancer dataset, suggesting they represent large regulatory domains composed of many smaller annotated elements. The example shown here is a 42 kb region on chromosome 4, which aligns with 5 different enhancer resources. And this demonstrates that our sliding window approach plus the merging strategy can predict enhancer regions of any length and can recover extended enhancer architectures that conventional methods often miss. Now, having validated the robustness of our model, we next examined how genetic variations might functionally disturb these critical regulatory regions, potentially influencing the disease-relevant gene regulations. First, we started by collecting the full DBSNP catalog and extracted all the short variants, which are less than 50 base pair, that fell in the screen enhancer regions or in matched non-enhancer flanking regions. This gave us the candidate sites where a single nucleotide substitution could potentially alter regulatory activity. Second, for each region, we substituted the alleles into sequence and reevaluated the enhancer probability using our DNABERT-Enhancer 350 model. And for transcription factor binding site within these regions, enhancer regions, we repeated this allele substitution framework using Deep vRegulome, which contained hundreds of DNABERT models trained on ENCODE ChIP-seq data. This step led us to identify variants that directly disrupt the transcription factor motifs and providing mechanistic insight into how enhancer function might change. And finally, we quantified the strength of this effect using probability differences, which is also termed as chord change, and also the log odds ratio for hypothesis. This allowed us to quantify whether a variant caused a loss of function, meaning reduced enhancer probability, or a gain of function where the enhancer like activity increased compared to the reference. Further, we applied empirical FTR threshold to highlight only the most confidence loss of function and gain of function variants. Now, to understand the biological and clinical relevance, we overlap these high confidence variant with clinical annotations available in ClinVar, GWAS catalog, and GTEx EQTL dataset. This analysis represents the core of our project, leveraging AI to systematically identify non-coding variants that actively reshape enhancer function, thereby revealing hidden drivers of disease risk and potential therapeutic targets. Our model predicts several candidate loss of function variants. We applied FTR threshold to the log odds ratio distribution and defined the empirical p-values. And with a cutoff of p-value less than 0.05, we identified significant candidate loss of function variants with high impact on regulatory regions. To connect these predictions to biology and disease, we then overlapped the loss of function variants with ClinVar, GWAS, and GTEx EQTLs and identified with the cutoff 8 of them as a clinically significant or with reported significance, adding an important layer of clinical grounding to the prediction. Now, the transcription factor analysis is where the things became particularly compelling. Among the variants with p-value below 0.05, we see strong distribution in a focused set of transcription factors. Displayed here is the top 20 TFs, which frequently disrupted in the enhancer region by these loss of function variants. These include major regulators like ATF3, ATF7, GATA2, etc., and the chromatin modelers, and as well as core cohesion components. Many of these descriptions shows extremely high loss of function with high log odds ratio values, which indicate the dramatic changes in enhancer activity. So the variants we identified aren't simply altering the model prediction, they may profoundly shift transcriptional program, and this gives us a mechanistic explanation of how enhancer loss of functions variants may contribute to disease-relevant regulatory dysregulation. Similarly, we identified several gain-of-function variants as well. We observed certain gain-of-function SNPs within enhancer regions which strengthen their existing activity and boosting the enhancer probability. And again, these high confidence variants intersected with GWAS and EQTL loci, suggesting they can modulate disease-relevant transcription. We also identified several gain-of-function SNPs in flanking enhancer regions that create entirely new enhancer-like sites, and these de novo enhancers may expand the regulatory landscape. Importantly, some of these newly formed enhancers also overlap with the disease-associated loci, showing that enhancer gain is a mechanism by which non-coding variants can impact gene regulation. Highlighting here is the ISM plot of an intronic variant in chromosome 19, which causes a loss of function in the enhancer region and disrupt the transcription factor binding site of MAX protein. On querying in GWAS, the variant is mapped to PCA-T19, a gene, and this variant is identified as a risk variant in prostate cancer. In EQTL analysis, the same variant is reported to influence the expression of PCA-T19 gene in multiple tissues, including the testes. The impact can be seen from this plot, which displays the shift in the high attention score of mutated region compared to the wild type sequence. This plot over here highlights the high attention scores of the given region. And based on the score change and log odds ratio, you can see how the high attention score changes comparing to the wild type and comparing to the, and how when the allele is changed to A or mutated, how the heat map changes. Similarly for the log odds ratio here. This is another example of a variant in chromosome 2. This is also an intronic variant, which causes a loss of function in the enhancer region. And this disrupts the transcription factor binding site of ATF4 in that enhancer region. And on analyzing the clinical annotation reported in publicly available datasets, especially in GWAS, the variant is mapped to HS1BP3 gene and is associated with arterial fibrillation. And the impact can be seen from the plot. Similar to the previous example, there is a shift in the high attention region compared to the wild type when the mutation is there or the mutated allele is present. The whole attention changes from that region and thereby losing the function. It is also interesting to note that all the candidate variants predicted by our models are in or around the high attention regions, and believe that all predicted variants present in the study provide strong candidates for further experimental testing to confirm their functional impact on a specific trait or disease. Now, I'll talk about the key findings one more time. In terms of performance and model insight, DNABERT-Enhancer 350 model performed better than 300, 201 base pair model, indicating that the model learns more context information from the longer sequences than the shorter ones, and they precisely locate enhancer regions while outperforming recent state-of-the-art method. In order to identify or validate the robustness of our method, we applied the best model, that is the 350 model, to human genome for a genome-wide enhancer prediction, and we identified approximately 1.82 million enhancers covering 21% of the genome. And while validating the prediction with the external databases, we found that 92% are validated by the enhancers present in even the high confidence databases or even from the databases which has prediction results. Coming to the biological and clinical relevance of the study, we identified several loss of function variants which causes a significant disruption in the enhancer region, as well as the core transcription binding sites within those regions, and they profoundly shift the transcriptional programs. Similarly, there are gain of function, which strengthens existing enhancers by increasing their prediction probability, and also create de novo enhancers in the matched flanking regions from the genome around the enhancer regions. And we also found that both of these variants overlap with the already available clinical annotations in ClinVar, GWAS, and EQTLs. And in terms of structural and functional insights, the sliding window approach, while creating the human genome for prediction, and along with the merging strategy of this merging of consecutive enhancer region of once a prediction is done, identified large enhancer domains and even enhancers with varying length. So even if the input is like a enhancer of any length, we can do a sliding window approach, and model can handle it and predict regions of any length, which usually the normal models often misses. And the model attention captures enhancer complexity without relying on any fixed positions, and they reveal novel regulatory architecture. And the future directions we are working currently on is to identify how the SNPs causes the loss of function or gain of function. We wanted to systematically analyze the somatic mutations in these regions and link that to the regulatory changes with disease phenotypes. Also, we'd like to do a CRISPR-based validation, the experimental uh, validation of the loss of and gain of function enhancer various to confirm their regulatory impact, and an ASO modulation like to target enhancer RNAs or key transcription factor transcript to fine-tune enhancer activity at the RNA level. And that concludes my talk here. And I would like to thank my Professor Dr. Ramana V. Davuluri for all the support and guidance I got for this project, and Dr. Farhad I. He's an associate professor at University of California for all the discussion and advices, and also Dr. Pratik Datta. He's senior research scientist in our lab. He's also co-author of this paper, and all the lab members, and also Northwestern Medicine Healthcare AI Forum for giving me an opportunity to present here, and the NIH funding support as well. And our paper is currently under review, but it is up in bioRxiv, and all the model is available in GitHub, and also all the predictions we are made it into a Streamlit app, which is not currently publicly available, but we will make it available once the paper is out. And you can, if you have any further questions or anything, you can contact me through my email ID. Thank you.
Rekha, absolutely wonderful presentation. This was a fantastic initial introduction with overview for people who aren't quite as familiar and in this space to bring them up to speed. And then how things opened up in the course of your talk is just amazing. This can fuel, this sort of research can fuel whole additional areas essentially of medicine, and for people to investigate how these newly identified enhancers can actually potentially assist with either rare diseases, diseases without treatments currently effectively, and with with this, so one can imagine, several years from now looking back and how this led to so much additional research. So just amazing, wonderful work with taking advantage of our newly available language model. Not that new, but applying it in this context to understand the genome in ways we couldn't otherwise understand. The question and answer is open, so please feel free to post questions. I just have a question here for a moment. If one were interested, for example, in a rare disease that has no effective treatments at the moment, but you did have potentially some SNPs identified for it, is there a way in which this, like, what would be your thoughts? I know it's going a little beyond some of the techniques, and I'll wait for some other questions about some of the specific techniques that you applied here. But are there thoughts that you would have if one had that in mind, how one might use your research?
If we have a set of SNPs available, my first thought will be like, if are these like mapping to any reported enhancer regions out there?
Not yet. They hadn't yet been reported to focus on an enhancer, but we have some SNPs in that regard.
Okay, then my if it is single nucleotide polymorphism, with our research, what we can do is we can take a region around that SNP, and extract that sequence, get a reference allele sequence, and introduce the mutation into the alternate sequence, and we can apply our model and do the prediction and see what the results coming out. That is the first primary thing we will be doing. And if we see that it is indeed, maybe since our model is for enhancer prediction, and we see that if it is showing a reference sequence, it is showing it is an enhancer region, and the model is showing any difference in the probability of the prediction, that means it can be a loss of function, and that region is like losing its function after introducing the mutation. So what we can do in such cases is we can do a CRISPR validation, then move to the experimental side and see whether it is actually causing any disruption to there. And just in case, if going a step behind, and if our reference sequence is not an enhancer, and this mutation introducing, after this mutation, we are seeing the probability has changed and the mutation is causing it to gain an enhancer there, then again, we will just move to the CRISPR validation and see whether it is actually performing. But if the region is not an enhancer region, then it will be a different question.
Right.
Yeah.
No, this is wonderful and can fuel tons of research.
So just to add to that, David, we can, if that is not an enhancer, we have several other models, like would it be affecting transcription vector binding sites or splice sites? So those things are, many things can be done with all these models. So like we are kind of exploring the dark matter in the genome, which has been underexplored until recently.
Thanks, Dr. Davuluri. We are starting to get some questions. Thanks for that slide. Also, we're getting some questions into the Q&A, so absolutely feel free to post. So this one from Joseph Peterson: is there a correlation between density of reported enhancers and length of predicted enhancer region from the BERT model?
I don't think so, because if you see the relation, the mutation can be like, as I said earlier, the length of the enhancer doesn't matter for this model. If you are getting a model, a region, like maybe, so the model is trained on 350 base pair. So if you are getting an enhancer region below 350 base pair, we can always flank the region and make it up to 350 base pair from the human genome, if you have the coordinate. And we can predict that using the model. And the SNPs can be introduced if it falls in within that region and do the prediction, get you the functional effect. If the region is a bigger region, more than 350 base pair, what we usually do is we apply a sliding window approach, where we divide the sequences, like overlapping sequences, like split it into multiple, and we do the same procedure and see what the effect of the SNP is. So regardless of the length, we can do the prediction. That is the one good thing about DNABERT-Enhancer. Yes.
Great. Thank you so much. There's another question more about when working with patients, for example, and applying this approach, are there additional legal implications from a consent basis for using this model or incorporating the findings of additional information provided by a patient?
First of all, I would like to clear this. So this is completely a decision support tool, not for the diagnostic purpose. So it helps researchers prioritize the hypothesis and everything. But any clinical use you have to do, it must go through the experimental validation. And I have stated this in our repository as well, like this portal is specifically designed for the research and exploratory use only. And the predictions are absolutely computational. So it should be interpreted as hypothesis generating and not for diagnostic or clinical evidence. I hope it clears.
Great. And we'll wait just another moment. It looks like someone is starting to author a question, but on your earlier slide, you had provided GitHub access, links to the papers, and the full recording and those links will also be available online as well for attendees here or people who had to leave the session early.
Yeah, but I would like we are not making it public so far, only for the reviewers, because the paper is under review. So once it is accepted and published, we will make it public for everyone. Yes.
Great. And then we have a more general question for someone who is interested in learning artificial intelligence in healthcare. Perhaps Rekha could additionally summarize how how AI specifically is helping her in this work. What aspects were the artificial intelligence in her work?
I think it more depends upon like what you want to study. The biological context is very important when it comes to AI. Like it depends upon what is your actual problem in the healthcare. So depends on that, you go through researches, understand the biology, understand your problem first, make sure you have like the data is available out there for whatever the purpose you are depending the AI on. And once you have the data, then you decide on what approach you want to do. Like there are so many artificially intelligent computational tools available out there. There are so many machine learning approaches out there. You can even go for a simple, if you have so many features and everything available for you to study your problem, you can just go for a simple model like a random forest. It's a simple one. But if you like go or want to learn more about like the context pattern or about the genome, human genome, sequence as an input or anything like identifying the regulatory regions or even transcription factor binding site, those kind of thing, or dependency on one region to another. So you will choose for a NLP based model to study. So that it all comes up to what you want or what is your problem.
What's your problem? What data you have available? What question you'd like to
Without
answer?
data, you cannot move forward. That is the main thing.
Exactly. And at Northwestern, I'll mention we are aggregating additional artificial intelligence resources for people who are interested in getting into this field who haven't yet jumped in. So thank you so much. Wonderful presentation again. Thank you for sharing your research with the community.
Thank you. Thank you so much for this opportunity. I really appreciate it.
Absolutely. And we're looking forward to seeing everyone back at the next AI forum.
Thank you.
Thanks.
Thank you.